It's a great pleasure to welcome you to today's webinar entitled WHO Fungal Priority Pathogen List, Challenges in Diagnosis and Management, organized by the International Society for Antimicrobial Chemotherapy and the European Society of Clinical Microbiology and Infectious Diseases. I am Maurizio Sanguinetti from Rome, full professor of microbiology and ESCMID past president, and it's my privilege to serve as one of today's moderators, together with Dr. Lass-Flörl, chair of the ISAC Fungal Infections Working Group. We are delighted to see such a wide audience interested in this important and timely topic. As many of you know, fungal infections represent an increasing serious threat to global health, a challenge made even more complex by the rise of fungal resistance. The WHO Fungal Priority List, published for the first time in 2022, marked a milestone in recognizing fungal disease as a major public health issue. This list identifies 19 fungal pathogens of greatest concern, prioritized according to their impact on human health, resistant patterns, and unmet diagnostic and therapeutic needs. It serves as a global framework to guide the research, surveillance, and policy efforts, helping to ensure that fungal infections receive the same level of attention and investment as bacterial and fungal threats. Today's session will help us reflect on the progress made since this publication, the ongoing diagnostic and management challenges, and the strategies needed to address emerging resistance. We have the privilege of hosting three fantastic experts, each of whom will shed light on specific aspects of fungal disease resistance. Professor Cornelia Lass-Flörl from Medical University of Innsbruck, Austria, will start by giving us an overview on fungal priority pathogens. Then Professor Sevtap Arikan-Akdagl from Hacettepe University Medical School, Turkey, will discuss Azole resistant Aspergillus fumigatus. And finally, Professor Anuradha Chowdhary from the Vallabhai Patel Chest Institute in India will explore the emergence and spread of resistant in Candida species. After the presentation, we will have a brief summary and an open Q&A question when we invite all participants to share questions and insights through the chat and the Q&A section. But please, we will have 25 minutes of presentation and 5 minutes of discussion after each presentation. So, without further ado, let's begin our session. It's now my pleasure to hand over to Dr. Lass-Flörl, who will introduce our first speaker, Professor Cornelia Lass-Flörl. Thank you.
Thanks, Maurizio, and hello everyone, and welcome to everybody. I'm Lass-Flörl, one of the microbiology consultants in Glasgow, and I'm the chair of the Fungal Infections Working Group of ISAC, and it's my pleasure to welcome Professor Cornelia Lass-Flörl, who is the director of the Institute of Hygiene and Medical Microbiology at the Medical University of Innsbruck in Austria. Since graduating from the Faculty of Medicine at Leopold Franzens University in 1986, Cornelia has specialized in medical microbiology. Professor Lass-Flörl is a fellow of several prestigious societies, including the American Academy of Microbiology, the ESCMID, the ECMM, and also the Infectious Diseases Society of America. Her research focuses on fungal infections in immunocompromised patients with special interest in epidemiology, diagnosis, prevention, therapy, and also antifungal susceptibility testing. Professor Lass-Flörl has published extensively and was listed among Clarivate Analytics' highly cited researchers in 2020, 2022, and also 2023. She has served in leadership roles within ESCMID, UCAST, the EFISC, and also the European Confederation of Medical Microbiology. So I now hand over to Professor Lass-Flörl for her talk, which is on overview of the fungal priority pathogens. Cornelia.
Yeah, thank you. Yeah, thank you very much. So good morning, everybody outside of my room. So for me, it's a great pleasure to give an overview of the fungal priority pathogens and let's say the practical challenges we face in diagnosing and management this infection. I will cover the most important drawn by recent WHO assessments and give a short summary in my slides. Due to time reasons, I can't go through all of the details, but I briefly highlight the most important points. So as you can see here, WHO categorized fungal pathogens into critical group, high group, and medium group. And I will briefly give a snapshot and discuss only what I think is very important. So finally, what I can summarize is that most of these fungal diseases, they span a wide spectrum from allergy to colonization to infectious diseases. And most pathogens, which you see here on my screen, are environmentally acquired, which does, with other words, mean that it's a local epidemiology, or that local epidemiology may differ from country to country. Usually, for all these pathogens, human-to-human transmission is uncommon, but still we lack robust data all over the world. And for some, we have whatever we call clinical breakpoints. And reliable broad access to antifungals all over the world is not supported right now. So directly jump into Cryptococcus neoformans. So usually what is important, having Crypto neoformans in mind. So usually the uptake of the fungus is via the respiratory route, and it's definitively inhaled from the environment. So there's a strong influence on the soil or on the environment during people living. In most cases, affected is either CNS or the lungs. And for some patients, specifically those who are slightly immunosuppressed, are associated with high mortality despite antifungal therapy. Risk factors are typical HIV and iatrogenic immunosuppression. As already mentioned, there is no human-to-human transmission. Hence, it is taken up by the environment. Preventability is moderate. And up to now, we don't have a vaccine. We can use prophylactic and preemptive therapy for at-risk group. And this is shown by several upcoming studies or studies which were done that reduces the incidence of cryptococcal meningitis. The opportunity when it comes to cryptoneoformans is that an early diagnosis will improve the outcome. And presently we have effective, fast, and cheap tests such as the lateral flow tests available. What is poorly understood up to now is antifungal resistance. We have a clinical breakpoint for amphotericin B, and there is some data that in not all cases fluconazole is could be associated with clinical failure. But what we need more data when it comes to Cryptococcus neoformans. When we move over to Cryptococcus gattii, then let's summarize, is also globally distributed, although this pathogen is more frequently found in tropical and subtropical areas. But as neoformans, soil and certain trees seems to be the source of infection. What is evident for Cryptococcus gattii? That it can adapt to different temperature settings. The overall annual incidence seems to be low, although gattii is associated with outbreak, and it seems that we have some endemic areas such as Australia and US, and they show higher rates of infection. Mortality ranges in between 20 to 25%, and usually immunocompromised individuals are at higher risk. But, and this is very typical for GATI, it also can affect healthy individuals. Going back, what are the typical risk factors? This is being critically ill or being immunocompromised or having an older age. The good thing is that Cryptococcus gatidis, there is no human-to-human transmission, but on the other side, preventability is low and there is no vaccine available. Resistance seems to be low, although we do not know that much. And all over, Cryptococcus gatidis accounts for at least in between, wherever in the world you are, in between 11 to 33 cryptococcal infections. Moving to Candida. As Candida is the topic by Suha, I don't want to step into very deeply. I just will comment on those pathogens from the WHO list. All over, Candida also spans mucosal disease, such as oropharyngale or vulvovaginitis, to cutaneous infection, to invasive candidemia, or to deep-seated infection. The first step, it seems, for the majority of the yeast, that endogenous colonization is de facto the first setup to have an infection, but this is not valid for all the Candida yeasts. Who is typical at risk? So typical at risk are those patients who are in the ICU, those with central-venous catheters, recent abdominal surgery, broad-spectrum antibiotics, and parenteral nutrition. So if we have a closer look to the priority list, 5 yeasts are categorized. This is albicans, this is Candida parapsilosis, this is Candida glabrata, this is Candida tropicalis, and this is Candida auris. So Candida albicans is probably the most important one because more than 90% of the human beings are colonized. And the good thing is that resistance is rare. Candida parapsilosis is listed simply because presently it seems that there is a clone spreading, a clone which is fluconazole resistant against parapsilosis. And in some centers, specifically in the south regions of Europe, this clone is associated with outbreaks and, let's say, faces a real clinical problem. So this Candida parapsilosis species, fluconazole resistant, is prevalent in more South Europe and South Africa. The other is Candida glabrata. Candida glabrata is potentially listed simply because it rapidly acquires resistance to echinocandins, and it already shows a high frequency of azole resistance. Followed by Candida tropicalis, which is mainly prevalent in Turkey, Spain, and Algeria, is also associated with horizontally transferred or horizontally spread in hospitals, and is widely distributed also in the environment. And as the queen, let's say the queen of all those typical species is Candida auris, which is well known now, such as a yeast behaving similar to bacteria, similar to MRSA, for example. It's a skin colonizer, and we have different clades with very different resistant patterns, and also associated, these clades are also with a different virulence. Clades 2 and 4 are rarely associated, for example, with invasive infection. And they are rapidly now becoming a major pathogen causing infection, for example, in the ICUs. Moving away from the yeasts to the molds, and typically the molds are uptaken also by the environment. Number one and most important, according to the WHO list, is Aspergillus fumigatus. So as Sevtap will give a presentation on azole resistance Aspergillus fumigatus, I do not want to step into, with the exception that azole resistance is emerging. But for me, when I step into the literature, it's obvious that there is a strong differences in local epidemiology. For example, from the country where I come, Austria, we have not found any azole-resistant strains up to now. It is a little bit in association that the widespread use of antifungals or azole fungicides in agriculture is contributing to the rise in azole resistant in humans. But for the moment, Sevtap will give more insights here. So let's move over to the next large, different group of fungi, which are the Mucorales. So the Mucorales are a group of fungi consisting of various different genera, such as Mucor, Lichthymia, or Rhizobus. And what is valid for Aspergillus is also valid for the Mucorales. They can make a wide spectrum of infections. But what is evident when it comes to Mucorales, they are rapidly evolving. And particularly, we have two different patients groups who are at risk. Those are typically the immunocompromised patients. In many, many cases, those are under voriconazole prophylaxis or under voriconazole treatment, because voriconazole does not cover the Mucorales. And the second patient population are those with an uncontrolled diabetes mellitus, and those with particular skin and soft tissue injuries. Invasive diseases is associated with a very, very high mortality, and it's really rapidly evolving. The good thing is that there is no human-to-human transmission so far, but on the other side, as it is dependent on the local spread of the environment, preventability is low. The only thing is we can't detect it as early as possible to improve survival, and still we don't have a vaccine up to now. Antifungal resistance, to be honest, is difficult to determine. We don't have clinical breakpoints available when it comes to the huge group of Mucorales. But what is important to know is that they are inherently resistant to fluconazole, voriconazole, and the echinocandins. The treatment of choice up to now is amphotericin B plus surgery. Another huge group are Fusarium species. This is also a large genus of globally distributed fungi, and usually they occur mostly in tropical regions, and they are also predominantly found in soil or organic matter and plants. Pathogenic for the human typically are Fusarium solani, Fusarium oxysporum, or the others. Invasive fusariosis is a life-threatening disease, and is associated with a very, very high mortality rate, up to 67%. Uptake is via the respiratory system or via direct inoculation, for example, for an eye infection. But depending on the immune status, it also can disseminate to CNS or other organs. What is important to know that Fusarium can cause fungemia, so a blood culture could be positive. The good thing is that there is no human-to-human transmission. Bad thing is preventability is low, and we do not have a vaccine available. Antifungal resistance is high, And it seems that Fusarium is inherently resistant to most antifungal agents, although we don't have clinical breakpoints established. Treatment of choice nowadays is voriconazole. The third most important mold group is Scedosporium. Also, Scedosporium is globally distributed, and typical infections are caused by Scedosporium boydii, Scedosporium apiospermum, or Scedosporium aurantiacum. Those are the most important from a clinical point of view. Invasive disease is also life-threatening and has very, very high mortality rates. Uptake is in the majority of cases via the respiratory system, inhalation, but also traumatic inoculation. Risk factors typically include those patients with hematological malignancies or other severe infections. Good thing is no human-to-human transmission. The drawback, once more, is that preventability is low, and up to now there is no vaccine available. Mortality is high, as already mentioned, and antifungal resistance is also high, but we do not have clinical breakpoints available up to now. Lomentospora prolificans is also globally distributed, can cause invasive diseases, and mainly affects immunocompromised patients, and is present in hot and arid countries. Is also a life-threatening infection with really high mortality rates, up to 70%. And the good thing is there is no human-to-human transmission. What is valid for most molds, preventability is low. It depends on where the uptake of the fungus in the environment is, and we do not have a vaccine available. Treatment is threatened by high resistance rate, and up to now in discussion is surgery, the usage of voriconazole plus minus terbinafine. There is maybe some light at the end of the tunnel. We have two more new drugs, which is fosmanogepix and olorofim, and the question is whether they will improve outcome of these life-threatening infections. Up to now, nor for fosmanogepix nor for olorofim, we typically have a clinical breakpoint. As another pathogen is Thalaromyces marneffii, is also a mold acquired through the respiratory route from the environment. And the typical source is decaying wood, for example. It's a dimorphic pathogenous fungus, which is endemic in South Asia and in some areas of China. In particular, patients with HIV as are typical at risk for infection, but also some healthy individuals can become infected, albeit to a much lower incidence rate. The treatment available, which is liposomal amphotericin B, itraconazole or voriconazole, unfortunately are not available in all these countries where the fungus is predominant. The good thing is that there is no human-to-human transmission, and preventability is low once more, and we do not have a vaccine right now. Complication with Thalaromyces marneffii is respiratory failure. Antifungal resistance is low, as well as we don't know that much about, and we do not have any clinical breakpoints so far. As a next, moving over to dimorphic fungi or fungi which are associated with the question, there is a pneumonia, but is not getting better with antibiotics, or some fungal infection can feel like a flu. And let's see what behind. So this is Histoplasma capsulatum. Histoplasma in general is globally distributed in the environment. Typical sources is soil, bird and bat droppings. Endemic regions is Latin America and Africa. We don't have that much study that we can really get more knowledge and annual incidence rates. But what we know is that once being affected immunosuppressed patient, dissemination may be active. And in some patients also healthy individuals can be infected. And typical, the first organ affected are the lungs. Histoplasma have the potential to produce outbreaks, but the good thing is that there is no human-to-human transmission, and depending on wherever they live, most people who inhale histoplasma do not get ill. Preventability is low, and up to now we don't have a vaccine available. It seems to be that antifungal resistance is moderate, but up to now we do not have breakpoints for antifungal resistance. Studies are very limited, but it seems so far that the MICs for Azole and amphotericin B are low. Cocci. I think this is a very important and very interesting pathogen. So the question is why? Because it's some of the most virulent fungal pathogens, is acquired also through respiratory, to the respiratory tract, simply because the fungus is present in the environment, mainly in arid zones in America, and the disease is also called the valley fever. Cocci may infect healthy individuals as well as immunosuppressed patients, and it's actually that fungus which can which is associated with the so-called flu-like symptoms. Risk factors are typical people of African descent, including African Americans, and being exposed to environmental dust and soil. What is interesting is that invasive coccidioidomycosis mortality is ranging into between 2 to 13%, but preventability is low as this fungus is in the environment. Up to now, we do not have a vaccine available. There is no human-to-human transmission, but what's going on right now, it is the discussion that it is believed that the climate change is responsible that in 2024 in California, they had an increase of this infection of 62%. So this is a really hot topic for those Americans living in these areas. Disseminated coccidioidomycosis is treated with fluconazole, itraconazole, or amphotericin B. Antifungal resistance is of concern. High MICs are found for fluconazole, lower MICs for the other azoles, but still we do not have clinical breakpoints available. Into this group, we need also to discuss shortly Paracoccides, is also found in the environment, has a medium mortality despite antifungal therapy, is endemic in Central and South America, is also found in the soil and mainly in the rainforest. But also the good thing is that most people are infected, they never develop symptoms, and there is no human-to-human transmission, but still preventability is low. And now move over to, let's say, an outliner of whatever I have told you right now, because pneumocystis jiovecii, the source of infection is the carrier or the patient suffering from pneumocystis infection. So it's clearly that there is a transmission from human to human, and being exposed can finally, depending on either an infection or no infection. So this makes the Pneumocystis jirovecii from the complete list of WHO gives a very special position. Although this fungal pathogen is globally distributed, the pneumonia is life-threatening, and outcome highly depends on the immune status of the person being infected. In majority, patients can be carried by healthy individuals and they don't have symptoms. So there is this human-to-human transmission possible. Although, so far as I understand, there is not really a cutoff given of how many pneumonia cells must be in the lung being at risk for infection. So those with HIV or organ transplant at highest as risk. So we can prevent by using prophylaxis. Trimethoprim sulfamethoxam is the drug of choice, and there is success, so it is preventable, but there is no vaccine available. Antifungal resistance is unknown, and we cannot use the phenotypic testing because we can't use all these culture techniques for pneumocystis. And the treatments we use are completely different as we use for fungal infections. So this brings me to my end. So first of all, putting together this WHO critical list where I just picked out what is important. In many cases, we don't have really good surveillance data. So we need to invest in surveillance, and we need to better understand epidemiology to close all these data gaps. We need to expand our knowledge how to best treat this infection, as well as we need to get more knowledge what are the best tests. Understanding the outbreaks supports a better prevention for other patients. Still, we have, it seems, high resistance rate, probably not necessarily always in vitro, but in vivo, and we lack a lot of breakpoints. And I think the real challenge is to strengthen our antifungal stewardship in the hospital, and to understand more which drugs are the best in treatment for these specific fungal pathogens. Thank you very much.
Thank you, Professor Lass-Flörl, for an excellent presentation. In the interest of time, I'm going to allow only one question. And the question for you is, what are the barriers for standardizing antifungal breakpoints for Candida auris, Candidozyma auris?
Sorry, I did not understand.
What are the barriers? What are the challenges for standardizing the antifungal breakpoints for Candidozyma auris?
Well, I think getting epidemiological cutoff is not the problem. The clinical cases are the problem. First of all, because coming up with clinical breakpoints, we need an in vitro, in vivo outcome. And as these strains are colonizers, as well as we have these different clades, I think we lack this data presently. And I'm not that sure, to be honest, whether the methods we use in detecting really are present whatever we have in vivo. So I think there is a little bit improvement, probably what is needed for the specific strains tested.
Okay, thank you very much. We'll come back to some questions after the end of all speakers, after all speakers present their data. So over to Maurizio.
Thank you very much. Thank you very much, Cornelia. Very, very nice presentation. So now it's time to move to the second speaker. It's a very pleasure for me to introduce Professor Sevtap Arikan-Akdagl. She's a great friend. I'm very happy to introduce her. Sevtap is director of the mycology laboratory at Hachet University Medical School in Ankara, Turkey, and she earned her MD and specialist degree in infectious disease and clinical microbiology from the same institution and completed a research fellowship at the University of Texas Medical School. She serves as secretary of ESCMID Fungal Infection Study Group, is a member of the UCAS subcommittee of antifungal susceptibility testing, and represents the ISAC in the Turkish Medical Society of Medical Mycology. She has contributed to ESCMID and team program committees, and she is an editor of antimicrobial agents and chemotherapy. Her research focuses on antifungal drugs, resistance, susceptibility testing, and the diagnosing of invasive fungal infections. So the talk of Sevtap is about a very concerning problem, the Azole-resistant Aspergillus fumigatus in clinics and environment, from screening to containment policy. So please, Sevtap, the floor is yours.
And this is for my disclosures, and I would like to start with some background information. Connie provided a very nice summary for the pathogens listed in WHO Fungal Priority Pathogens List. And Aspergillus fumigatus, which I will be focusing on, is in the critical priority group. It's in general the most common cause of invasive mold infections. There might be some changes depending on the host, the underlying disease or the clinical picture, but in general, it's the most common mold causing invasive infections. And actually, it's the only Aspergillus species included in this list. So if we go into more detail why it's in the list, the major problem is the ongoing variability in resistance rates to triazoles, increase in these rates, and also the cross-resistance among the azoles, also involving isovucanazole, which is the most recently introduced one. So while WHO fungal priority pathogens list includes Aspergillus fumigatus, for the CDC, it's in the watch list again, so it's gaining more and more importance. Now let's talk about intrinsic and acquired resistance in Aspergillus fumigatus in broad sense as the species complex and also as sensu stricto. Antifungal resistance actually started to draw attention in 1970s, but it's by 1990s when azole resistance emerged, and it's actually 1997 when the first azole resistant, actually itraconazole resistant Aspergillus fumigatus strains were reported. So this is the list for the essential antifungal agents. And we have resistance concerns for almost all, I would say, but the degree of the concern is different. And for itraconazole and voriconazole, the rising problem again is Aspergillus fumigatus, together with some other species. Intrinsic resistance, to start with. Actually, intrinsic resistance clinically is observed as breakthrough mold infections. And for Aspergillus genus as a whole, including all species, the fluconazole intrinsic resistance is a very well-known entity. If we go further with some cryptic species, and if we focus on section fumigate only, for Aspergillus fumigate affinis, for Aspergillus lentulus, and Aspergillus udagave, we might have the problem of reduced susceptibility or resistance for the azole. So this is the intrinsically observed pattern. What about secondary or acquired azole resistance? How do we detect it, and what are the molecular mechanisms? First of all, in vitro resistance is only one of the factors that influence antifungal efficacy. And the way to show it for an individual strain is by performing antifungal susceptibility testing. But many other factors of the host, the immune status in major, and of the antifungal drug may play a role in clinical outcome. So in other words, refractoriness and resistance are not the same for invasive aspergillosis and for also any other infection. So natural or acquired resistance is only one of the fungal factors that may lead to a refractory infection. So how to detect antifungal resistance for Aspergillus? We have the reference CLSI and UCAS microdilution methodologies. In addition, UCAS also developed an agar screening assay as exemplified by Aspergillus fumigatus. This has been practically very useful because microdilution assays are labor-intensive, need expertise, and may not be implemented in every lab. In contrary, this agar screening method may be easily applied in microbiology laboratories, and if a strain comes out to be resistant with the screening assay, it may be confirmed at a reference lab by microdilution methodology. And this is actually a UCAS document that has the screening method for the detection of azole and echinocandin resistance. And you can see the design of the plate. It includes itraconazole, voriconazole, posaconazole, and the growth control wells. So the molecular mechanisms of resistance. Different mechanisms may be working and they can add up on top of each other actually too. CYP51 amino acid substitutions is one of the major mechanisms. Another one is the overexpression and the tandem repeats, which are important, especially in the environmentally acquired resistant strains that I'll come to later. And also the efflux pumps. Efflux pump overexpressions may lead to azole resistance and some other mechanisms, for example, biofilms. Today we know that Aspergillus can also produce biofilms, which render resistance to antifungal drugs. And if we know the mutation, we can predict the resistance phenotype. Here is the list, an example list actually, and if I just go as an example through one of the common environmental changes, which is the tandem repeat 34 together with L98H, itraconazole and posaconazole resistance is almost always there, but for voriconazole and isoviconazole, variable susceptibilities are obtained. Azole resistance in Aspergillus fumigatus and One Health perspective. So azole resistance is important in human health, in animal health, and also in environmental health, in agriculture and horticulture. So this has some reflections, certainly. We have two main resistance selection routes. The first one is the de novo route, which means the azole therapy leading to development of resistance. This is mostly observed in patients with chronic pulmonary aspergillosis receiving azole treatment for long, though we do not exactly know the duration needed for development of resistance. The second major route is the environmental route. Here, the demethylase inhibitor group of fungicide use is important for plant protection. At the very end, an azole naive patient may end up with a resistant strain, which he acquires from the environment. And within this context, also the agricultural products are important for national and international trade. There are also studies performed in this respect, the soil and plant bulb samples, and it's possible to show the genetic relatedness in different countries. There are some authorizations and regulations for the use of this DMI group of fungicides. And here is an example. You can see different types of azole compounds that are used as fungicides. And actually, tebuconazole is one of the most commonly known one. And in some parts of the world, in some countries, in United States, triazole fungicide use has been reported to get increased after 2006 and to 2016. So this is an important issue, and certain regulations and authorizations are needed to follow the use of these compounds. Why is it important to have these azole fungicides in the environment? Well, because they have low biodegradability and they're easily transported into the environment, and they become persistent in soil and in water. So you can have traces of these on fruits, vegetables, on flowers, and can just carry them to home. And drinking and irrigation water can also get contaminated. I'll come to that a few seconds later. For rains, one can think that it's good to have rains since these can dilute the fungicides, but if at sub-inhibitory concentrations, this can also contribute to emergence of resistance by stimulating, for instance, overexpression of the efflux pumps. Here is the European Environment Agency limits for the pesticides in European rivers, lakes, and groundwaters. As you can see, two azoles, epoxiconazole and propiconazole, have also been included, and the rates of exceedance and the threshold used for calculation. Another one, the European Union drinking water regulations. You can see the pesticides in total. So you can have an idea if there has been any excess amount in general. So sampling is important for environmental surveillance. For the sampling strategies, what is in general implemented is the air sampling. First of all, the volumetric one is preferred because it is quantitative, or air sampling by settling plates, which will provide semi-quantitative data. And with swabs for surfaces, also using contact plates like the Rodak plates, that might also give an idea about the existence of the resistant fungi. Also, due to some, for instance, seasonal spore peaks, or during constructions due to increase in dust, some adaptive strategies may also be applied, and some other surveillance studies might be performed. And this is an example for the workflow for azole-resistant Aspergillus fumigatus surveillance in a hospital. For this environmental surveillance, weekly air sampling is recommended, and duration time might be increased depending on the yield actually. Might be 3 minutes to start with, but that might be extended to be done weekly. If there are increases in the number of the patients with aspergillosis or in air sampling, then swab sampling should also be performed in the hospital. And if the resistance rate is more than 10%, sampling surrounding the hospital is also recommended. So after these samplings, these should be inoculated and subcultured. Some specific media may also be used, such as the selective flamingo medium to suppress the growth of order Mucorales, and by incubation at 48 degrees Celsius. And finally, after the growth and doing the susceptibility testing, the resistance rate may be calculated monthly or every 3 months. Birds are also important in terms of azole resistance in Aspergillus, because they may disperse these resistant strains. And actually, there are some studies focusing on the migratory routes of birds and the genetic relatedness depending on this. Birds can also get infected with Aspergillus, especially wild birds. And there are some systematic reviews for the animal aspergillosis as well, including the articles addressing azole resistance in animals and immediate environments, including also descriptive clinical cases in different animals. In clinical practice, itraconazole is most common treatment for cats, dogs, and horses, and voriconazole for zoo animals and wildlife. And you can see the number of the articles that have been encountered in this systematic review from different geographic regions. Finally, the conclusion is that non-wild type Aspergillus fumigatus is a more significant concern for zoo animals, wildlife, horses, and poultry than for other animal categories, for cats and dogs. Very recently, European Food Safety Agency published a report. This was in collaboration with the European Centre for Disease Prevention and Control, European Chemicals Agency, the European Environment Agency, the European Medicines Agency, and with the support of the European Commission's Joint Research Centre. This is a very critical and important initiative. This is for the first time, actually, such a multi-agency collaboration. And this report focuses on the network activity, including the academic, the industry, and the policymakers to build roadmaps to reduce the threat of antifungal resistance. And actually, what are outlined in the report are shown here. The data on azole usage and monitoring, this is missing in many regions and countries. Azole authorization and validation. The best practices is very important. The genomic epidemiology and the ways of transmission, the spread, and finally, all steps to reduce the risk of azole-resistant aspergillosis in humans as a One Health perspective. If we have a look at the resistance rates globally, it changes a lot, as Connie also has touched, both in clinical and environmental strains. And if I show a more recent review, again, we can see that the rates actually present with a mosaic of local epidemiologies. It might be 0% or it might be as high as 30%. So local surveillance studies are crucial. At this point, I would like to give some data from a multi-center study we conducted in Turkey, and with 2 coordinator centers, my institution, Hacettepe University, being one of them. 21 centers participated in the study. Clinical and environmental strains were studied. Not all centers provided both, but we had finally both of these groups of strains. And reference UCAST methods, both agar screening and microdilution, were used for MIC testing, finally, and confirmation of resistance. So what we had at the end, the resistance rate is around 3% for clinical and around 1% for environmental samples. There is a recent publication from the country suggesting a higher azole resistance rate, but no reference susceptibility testing was used in that study, so that needs to be confirmed. In addition to the low rates of resistance, so we also studied the CYP51A mutations, and close to 50% of the strains had the environmental related change, namely tandem repeat 34 combined with L98H. And for the rest, we could not detect any mechanism suggesting that some other mechanisms are working and needs to be worked out. And here are the samples which yielded resistant strains from different cities in the country. and these are the environmental ones from hospital, also agricultural soil. Some genomic and phenotypic studies are also being carried out. This study is from the Netherlands. And most importantly, mixed Aspergillus fumigatus genotypes might be isolated in one patient. So that is really a challenge for clinical management. So aspergillosis can develop in different patient groups, in immunosuppressed individuals, in ICU, but also in some patients with chronic pulmonary disease, such as cystic fibrosis. And this is a summary of the studies in this respect. If we have a look at the numbers, again, we can see that the rates vary and also the mutations vary. How do these in vitro resistance data translate to clinical practice? These are the in vitro in vivo correlation studies, and I know they're very small, but I can summarize them for you to conclude. And we can say that the resistance in Aspergillus fumigatus adds up to mortality to a remarkable extent. Some studies have not supported this conclusion, but those were of a low population size, and probably that's the reason. And in ESCMID ECMM-ERS guideline, there are recommendations related to intrinsic resistance, focusing on fumigatus complex, for instance, for Aspergillus lentulus to avoid azole monotherapy. For the local epidemiology, if the environmental resistance rate is 10% or more, the primary therapeutic choice, which is voriconazole, needs to be changed. And similarly, if azole resistance is documented, again, the treatment recommendations are different. This table is, again, from ESCMID ECMM-ERS guideline. And if a resistant strain is infecting the patient, instead of voriconazole, either liposomal amphotericin B monotherapy should be used with a strong recommendation, or voriconazole plus an echinocandin, anidulafungin in this case, is a moderate recommendation to be used for treatment. So to conclude with, our knowledge on intrinsic resistance is increasing, but the rate, what is increasing in terms of the rate is of the secondary resistance in Aspergillus fumigatus, at least in some centers and regions in the world. And in vitro azole resistance in fumigatus has impact on mortality. It increases mortality rates. So surveillance programs are very important. The approach should be with One Health measures. And we can clearly see that there are remarkable differences in terms of the rates of resistance. Crop fungicides are drawing much attention. Regulation and authorization is needed and is lacking in many countries and regions in the world. And the One Health perspective is very important for better outcomes in invasive aspergillosis due to resistant Aspergillus fumigatus. And with this, I would like to stop and thank you for your attention.
Thank you very much, Sevtap. A very, very comprehensive, nice, fantastic presentation. Thank you so much. So we have time only for one question. So I ask you if you have time to look at the question and answer section to reply to other questions you received, because you received several questions. The question is, what are the implications of intrinsic azole resistance in Aspergillus species on antifungal treatment outcomes? And how can these impact prophylactic and empirical treatment strategies?
For actually all of these treatment strategies, for prophylaxis, for empirical, and also for targeted treatment, if we use an azole compound and if there is intrinsic resistance of the infecting strain, there will be clinical failure to start with. And the other point here is the development of breakthrough infections. For instance, if we are using azole prophylaxis and if we have a strain from Aspergillus fumigatus complex, which is intrinsically resistant to that prophylactic agent, then a new infection might develop due to this species. So actually, this is what's usually happening in clinical practice. The breakthrough infections is usually the alarming point, saying that the infecting strain might be intrinsically resistant. So the recommendations already documented in guidelines are important in this respect. And we actually have a list getting longer and longer each day with the cryptic species being revealed with molecular studies. So a good knowledge and awareness regarding intrinsic resistance is needed in this respect.
Thank you very much. Thank you very much. So, Abs, is your time to present the last speaker. So, please, thank you.
Thank you, Maurizio, and it is my pleasure to introduce my friend, Anuradha. Anuradha Chowdhary is Professor of Medical Mycology at the VP Chest Institute at the University of Delhi, and she currently serves as the Secretary General of ISAC. Her research focuses on antifungal resistance, molecular epidemiology, and emerging fungal pathogens, particularly multidrug-resistant Candida auris and Cryptococcus, and also azole-resistant Aspergillus fumigatus. Anuradha has authored over 200 peer-reviewed publications and is a fellow of several scientific organizations, including serving on the ESCMID Global Scientific Program Committee. Over to you, Anuradha.
Thank you, Abhijit. Thank you so much, Abs, for such a kind introduction. And it is always difficult when you're a last speaker from, the fantastic speakers. I learned a lot from these two, presentations. And now I will be talking about emergence and spread of resistance in Candida species. And specifically, I'll be talking about the important drug-resistant pathogens in WHO priority pathogen list. So drug resistance in infectious diseases is a real important threat. And we all are aware of about global antibiotic surveillance report, which recently came in 2025, just a few days back, where we heard that 1 in 6 common bacterial infection in lab is resistant to antibiotics. And also we know that resistance has increased among the several antibiotics, but here I would like to emphasize, as usual, as always, antifungal resistance is also a serious public health threat, and it is largely ignored. But it should not be ignored. And why it should not be ignored? Because unlike bacteria, we have very limited classes of antifungals. And when we have antifungal resistance, we lose practically 2 or 3 classes of drugs which are used to treat patients. So why I'm saying that antifungal resistance is ignored, it's because only in 2019, we have the first fungal species, Candida auris, to be listed in the antibiotic resistance threat reports. And also just recently, 2022, that we have this fungal priority pathogen list. Before that, fungi as such didn't make to any of the WHO or other lists. And with that, I'm going to discuss today more the fungi or Candida species which are multi-drug resistant or which are azole resistant and which has potential to spread to the larger community or in the ICUs. With that, I would like to start with Candida auris. And we are aware that Candida auris was first isolated in 2009 from the external ear canal from a Japanese patient. And in the same year, several isolates, about 15 isolates, were also reported from the external otitis media cases from South Korea, but that time not labeled as auris. And it was only in 2013-14 that we had several reports which came from initially from Indian subcontinent, and which showed that a new clonal strain of multi-drug resistant Candida auris has emerged. And this strain was resistant to azoles as well as to other antifungals, specifically amphotericin B, followed by this report reports from South Africa and Venezuela appeared, and that's a time 10 years back in 2013 and 15. At that time, and also at present, we know that Candida auris is very close in the Methanovi complex, and this complex is known for antifungal resistance. And it also, this species is different than other Candida species. Why? Because it has not only resistant to multiple antifungals, but it persists in hospital settings and is associated with high mortality rate ranging from 30 to 60%. The epidemiology and initially the Candida auris infections were underreported, and that was highlighted in the initial studies where a lot of investigators, including our group, showed that the commercial identification system did not have this new fungal species at that time. And at present also, we face a lot of challenges with the phenotypic or biochemical identification systems to identify Candida auris. And also initially in the matrix in the Amaditov, the limited strain data were not able to identify several clades of Candida auris. But the last decade has witnessed tremendous improvement, and a lot of systems have included Candida auris. And now Candida auris can be easily identified by using Malditov and also with other commercial identification systems. And maybe that identification problem was associated with the wrong identification or less reports in the initial phases when Candida auris was identified. So till 2009, only few countries had Candida auris. But if you look at the last few years, you'll see the sharp increase in the number of countries which are reporting Candida auris, either as a single case or multiple outbreaks in the hospital settings. CRS presently has been reported for about 50 countries to date, and among all the continents have reported Candida auris as a very important threat. And if you look at the global map of Candida auris, it looks like the red, and red shows outbreaks. And outbreaks are primarily occurring in intensive care units in adult ICUs. You'll see a lot of outbreaks which have been reported from North America, from Indian subcontinent, and from Africa, as well as from the Latin America. So as the first speaker, Dr. Cornelia, said that Candida auris behaves like a gram-negative bacteria. And actually, the auris epidemiology is actually tied to, its epidemiology is based on the fact that it is transmitted from skin of the colonized patients, which shed the skin scales in the vicinity in the hospital environment, and from there it can be transmitted to another person. So this is very unlike of other Candida species like Candida glabrata or others. And because of persistent colonization of skin and abiotic surface, it contaminates the high-touch surfaces areas. It contaminates for a prolonged period of time. And porcine and human ex in vivo models have shown that C. auris can cause sustained colonization. And what is more important is that it forms multi-layer biofilms under human sweat conditions. And it resides deep in hair follicles and penetrate deep in skin layers. Therefore, it evades detection by routine routine swab cultures also sometimes. And the manifestation of C. auris, once it colonizes the patients, and once you have a breach of skin, it behaves similar as other Candida species causing candidemia. And it manifests similarly with other clinical conditions like urinary tract infections, wound infections, and also disseminated disease. The risk factor for colonization of invasive Candida auris infections are similar to other Candida species. And also the crude case fatality rate is similar, 30 to 60%. And this is one of the very important Candida auris specific addition which has been found, known as SCF1, surface association 1 factor, which has been labeled as very distinct and important addition found primarily in auris, which helps in making biofilm formation, which is also one of the virulence markers. And if you look at why not everyone gets colonized with Candida auris, because still IL-17 pathways, the dysbiosis which leads to IL-17 pathways dysregulation, helps in getting the colonization of the patients. And these pathways are still, several groups are working, but at least we know that the dysbiosis occurs in this pathway, which leads to the colonization of the patients. And now coming to the genomic epidemiology, this was the first initial paper where 19 countries data set was obtained and whole genome sequencing was done. And this was the paper which showed us clearly that initially 4 clades have been associated to Candida auris. And these clades were specific to the geographic locale. And if you look at the time to most recent common ancestor, they found out that Candida auris emerged about the last 400 years ago. And Candida clade 4, which emerged most recently, 30 to 40 years. And clade 2, which is the Japanese or South Korean strain clade, was the oldest one. And also, if you look at these study also identified chromosomal arrangements, other genetic markers, and found that generally the Japanese strain, which was isolated from external ear canal, was susceptible to fluconazole, and it is still found to be susceptible to fluconazole. And majority of resistance is found in clade 1, which is the Asian clade, and also amphotericin B resistance now is rising, initially reported to be 23% in this study. But if we look at recent data, it about 30 to 50% of the isolates are showing amphotericin B resistance, and echinocandin resistance is still less, which is about 5 to 7%. So auris is considered to be intrinsically resistant to azoles, fluconazole specifically. And this azole resistance occurs due to mutations in ERG11 gene, which is the azole target. And clade-specific mutations have been associated with the resistance. And also, if you look at, so far, the global transmission has been attributed to clade 1. But then, as the genome of larger isolates has been done from many centers, we are also witnessing new clades. So this was 5th Candida auris clade, which was identified in Iran. And more recently, clade 6, which is distinct from other clades, has been identified in Bangladesh as well as from Singapore. And if you look at the resistance mechanism, which underline resistance in Candida auris, the most important, I'm going to talk about fluconazole, because fluconazole is the first line of drug which we use still in low and middle income countries where access to echinocandin is limited. And here, as I said, that the specific azole ERG11 gene mutations are associated, where you can find out these mutations which are specific to one geographic locale. But in addition, also mutations in transcription factor has been associated, which causes in turn, increased efflux of the drugs. And coming to this is also a very important mechanism where, in addition to ERG11, this leads to resistance. And if you also see echinocandin resistance is worrisome because it develops while on therapy. And FKS1 hotspot mutations are known while on therapy, where echinocandin resistance is majoritarily reported from North America and also from other parts of the world. And these are the mutations which are specifically labeled for echinocandin resistance. And for amphotericin B, which is still very important in countries where echinocandin is not available. So the permeability changes in the cell membrane is basically responsible for the resistance in amphotericin B. And clade 1 isolates really show high resistance. But few reports have emerged which have shown that ERG3 and more recently ERG6 mutation in single isolates from clinical patient has resulted in high amphotericin B MICs up to more than 8 microgram. And if I come, why we need to study this antifungal resistance? Because once we have this antifungal resistance strain in one of our ICUs, so this resistant strain, or if it is echinocandin resistant, or if it is pan-drug resistant strain, it can be transmitted from one person to another person. So first of all, in 2019, this report came from CDC, where they identified in the United States the strains which were resistant to all the three classes of antifungals. And initially, by that time, no resistance or no transfer of these strains from one patient to another patient was observed. But later on, transmission of pan-resistant and echinocandin-resistant strains were observed, suggesting that once we have this strain in the hospital environment, it would be difficult to eradicate. It would be difficult to, control the spread of these bugs. And also this kind of transfer we also saw in COVID time. In COVID, this is the initial first study, the early pandemic, where we observed that multi-drug resistant strain of Candida auris was transmitted from critically ill patients from one person to another. And there was a high mortality of 70% in these patients. And if we, the epidemiology of Candida auris has been very well studied in the United States. And there, although it was just reported in 2016 in the United States, but initially the reports were because of the transmission from of the other clades by international travel. But later on, in last 4 or 5 years, their local transmission is happening in the hospitals. And if you look at this worsening spread of Candida auris in the United States report, you'll see that 44% increase in 2019 to 95% increase in the cases of clinical cases were observed. And not only that, the worrisome aspect was that echinocandin resistance, which increased from 2021. In 2021, it was about 3 times that of previous 2 years. And majority of these resistance is is found in facilities where acute skilled nursing facilities are there, where ventilator units are used. And it's about, after the acute care, long-term, these patients stays in these units, where if the surveillance is not up to mark, and then these strains can go from one patient to another patient, and these patients when shift to another hospital, and if the surveillance is not done there, these patients are source of infection in the other hospitals. And outside the hospital settings, Candida auris was not reported till 2019, and it was, as Dr. Cornelia mentioned, that cocci is one of the important pathogen which has been linked to global warming and, in the United States. And also Candida auris is one of the yeast pathogen which has been thought to emerge because of the climate change and warming climate. Why? Because Candida auris can grow at high temperature, 40 to 42 degrees, and can also grow in saline at higher concentrations. So it was believed in this hypothesis that this is the yeast which emerged because of the climate change, and probably it existed in coastal wetlands. And this was one of the first paper which confirmed that Candida auris existed in the coastal wetlands, and it was identified from sandy beaches with human activities, where it was probably a contamination from the people who were visiting the beach. But the most important part of the study was Candida auris was also isolated from very high salt marsh area with no human activity. And this strain was found from Andaman Island. And this strain was found to be growing very slow at 37 as well as 42, suggesting that it was still acclimatizing to the temperature. And also it has one copy of ERG11 gene as compared to the duplications which are found, because this isolate was susceptible to fluconazole as compared to the isolates which are found in the beachy areas. And after that, also it has been identified in Colombia, coastal habitat, and C. auris is now used, can be identified in wastewater, targeting wastewater surveillance. We can predict the outbreaks. And this is one of the studies where they have found in United States, where they saw that the areas where the wastewater surveillance was done from the hospitals, it could even detect by PCR the clade-specific detection in the wastewater surveillance, suggesting that this would be very helpful in settings settings where you could invest in wastewater surveillance. And outside also, it has been identified in the resistance has been linked to the fungicides which are used in the environment, as we heard in the previous talk by Dr. Sevtap. And in this study, we found that the surfaces of stored apples, which have a high quantity of fungicides, we found several distinct strains of Candida auris. And also these strains were resistant to tebuconazole. So probably in the areas where azole fungicides are used, auris, if it comes in contact, can also develop resistance to azoles. And what was interesting in the study was that One Health, these strains were related to not only to the patients or in our country, in Indian subcontinent, but also certain strains were related to the isolates from clade 1 from Canada and French isolates. And one of the patients had traveled in India before, when this strain was identified, suggesting that, the environment also very, very important in transmission of Candida auris. And finally, for Candida auris, screening is a very, very important tool in identifying and controlling new introductions of C. auris. And the most important step for prevention of this Candida auris, you'll find several guidelines, is the transmission-based precautions, which has to be followed. And also we know that it is resistant to several classes of even the compounds which are used in the hospital settings, the disinfectants, and the quaternary ammonia compounds, which are routinely used, are not effective against Candida auris. So with this, I would like to come to the other aspects of Candida species. And we saw this slide, and we know that Candida parapsilosis is a matter of concern. It behaves similarly like Candida auris. It's fluconazole resistant clonal strains, which has a tendency of outbreaks in several hospitals in Southern Europe and Africa. And then I'll also talk about Candida tropicalis, which is the second most important Candida species, first or second most important Candida species in Asia. And then I'll talk about Pichia kudrezii or Candida krusei. So if I come to Candida parapsilosis, Candida parapsilosis can be a second, if you look at this worldwide epidemiology of Candida parapsilosis, excellent review published in 2023 in Lancet Microbe, which shows that the Candida auris can be second to fourth most common Candida species, Candida parapsilosis, second to fourth most common species in ICUs, specifically also in pediatric ICUs. And also, if you look at countries like Brazil, like China, more than 20% of these parapsilosis is found in the ICUs. And if you look at the fluconazole resistant isolates, although some centers, it is just a single center based studies from many geographical regions, but a very high resistance rate has been observed. More than 30 to 40% have been observed in countries. And this is really worrisome, because once this resistance occurs, then as in these places, fluconazole is the most important drug, as I said earlier, because access to other echinocandins, and echinocandins are considered to be expensive in public sectors. So then it becomes difficult to have other classes of drugs to be used. And also, if you look at this data, so the reports of rising resistance of fluconazole in Candida parapsilosis emerged from as early as from 2018. And from 2018, the studies have gone high. And so far, many centers are reporting fluconazole resistant Candida parapsilosis with more than 10% of resistance rates. And this is one of the examples where excellent survey done by CDC, where they actually showed that 17% fluconazole resistance was observed in Candida parapsilosis isolates in 2020. And just in 1 year, this resistance rose to 34%. So this resistance rate is quite alarming. And also coming to not only the United States, but if we go to the low middle income countries, we'll see that the parapsilosis is causing concern in our pediatric ICUs. And in this pediatric ICUs, this is one of the study from Nilesh Govinder group, where they studied candidemia from 2012 to 17 in the hospitalized children in South Africa. So what was more concerning was both Peropsilosis and albicans. Peropsilosis topped the list, was most common species. And the high resistance, like 55%, was observed in fluconazole in Candida peropsilosis. And also, similar scenario was seen in Turkey, where in pediatric candidemia, peropsilosis was the #1 pathogen causing candidemia in pediatric patients. And this is our study, it's about 7-8 years back, but it was also quite alarming. This was my first study from India, which showed that 32% of the isolates were fluconazole resistant. And we also showed that novel mutation K143R was associated with fluconazole resistant in ARG11 gene. This was unlike the Y132F mutation, which is observed globally. And the Candida peropsilosis, this mutation is seen in both peropsilosis and tropicalis in several countries. And now coming to Candida tropicalis, very important agent of candidemia, adult ICUs in the Asia-Pacific region. Specifically in the Asia-Pacific, it is first or second most important, and it also the third most common species globally. And in the last 10 years, very high resistance has been reported to fluconazole. Specifically, the group from China has done a lot of work, and they have reported that 16 to as high as 30% resistance rate in China has been observed in the candidemia isolates in Candida tropicalis. And also Asia-Pacific, if this you see the century surveillance from 1997 to 2016, still the Asia-Pacific region had a highest resistance rate as compared to Europe, Latin America, and others. So just a brief note on other countries, scenarios in other countries. And this is one of the fascinating study from Algeria, where Dr. Cornelia is one of the authors in this study. And what this study showed that 7 hospitals were screened and in Algeria, and Candida tropicalis was the main cause of candidemia in 7 hospitals, and followed by Candida peropsilosis, both we covered today. And what was also important was shown that inter-hospital transmission occurred on 2 occasions for peropsilosis, and also fluconazole-resistant Candida tropicalis had a novel mutation, unlike the one which we showed, which is common globally. So this kind of global, the local epidemiology is very important to understand. And if you look at the fluconazole resistance in Turkey, and this study also highlighted the fact that the similar kind of mutations in Y132F were observed in the isolates. And just to quickly tell you that how one single clonal strain can emerge and can disseminate like Candida auris. So this is a study from China where they clearly showed that clade4 is associated with Azole resistance. And in the last 2012 to 16, they showed in China, this clone has rapidly emerged and has shown that this was due to one mutation which is occurring in the ARG11 gene Y132F, and due to tandem repeat. And this MLST clade4 has been observed not only in China, but also in our recent work, where we have more than 1,000 isolates. We did the surveillance from 2015 to 24 from 50 hospitals in India. We observed that about 5 to 6% of our isolates were fluconazole resistance, not as high as China. But the most important concerning point was that we also observed the similar mutation Y132F targeted in our patients, and which was causing the Azole resistance. And when we did the genome sequencing of large number of isolates and used the China isolates to see how our isolates fit in, we found the same clade which has emerged in the Indian subcontinent, MLST clade4. And these resistant isolates were distinct from China, but were close to China. And these are highly related, and we saw transmission in the ICUs of this resistant strain. So this would be, again, another important aspect in the Asia-Pacific region. And if we shift our epidemiology to Europe, and this is one of the recent studies from Netherlands, unlike Candida Azole resistance, 5FC resistance or 5FC non-wild type isolates have been emerged in Netherlands, and they have more than 40% isolates show resistant to 5-flu cytosine. And the non-wild type population had this specific mutation. So, the epidemiology varies, and continuous surveillance is very, very important. And this kind of 5FC resistance has also been observed in Denmark, in France. So if you look at Azole, all the 5FC is not used it's not used for treatment purposes, but except for meningitis, probably for cryptococcosis, but still this resistance which is occurring, and a clonal resistance which is occurring, is worrisome. And if you look at the the other studies where I will shift the focus to Candida glabrata, because Candida glabrata is considered to be a very important pathogen, and specifically pathogen important in North America, where resistance not only to fluconazole but also to echinocandins, and echinocandins are the treatment of choice in countries which can afford, like North America, Europe. So this Candida glabrata has been shown that this is one of the studies where 561 isolates were submitted to global surveillance, and from 2018 to 19 from 68 hospitals, and specifically from 28 countries. And if you look at the surveillance, majority from North America, 6.1% of isolates overall were resistant to fluconazole. And of these, 8.1% were from North America. And all fluconazole-resistant isolates were non-wild type for voriconazole. And also 2.3% isolates were resistant to at least one of the echinocandins, either Nidula, Caspor, mycofungin. And I'm not showing here, but I'm calling it as glabrata, but we are aware of the name change, taxonomy changes, and now it's called Nexomyces glabrata. And although this is not the topic of today's, but to be aware. And then I will also tell that although glabrata is reported to have resistant to fluconazole in North America, Europe, but this is our small study which we did from 10 hospitals, we did not find any rise in MICs in either in azoles or in echinocandins in our Indian study. But what was concerning was that we found defects in DNA mismatch repair. And when these isolates are exposed to azoles, they can develop resistance. So with this, I would also like to talk about the rare, but not so rare, because the strains, the species which are found as an outbreak pathogens and which are listed in our WHO list. This one is Pichia kudrezii, earlier known as Candida krusei. Pichia kudrezii is the most common species causing candidemia and hematological malignancies in North America and Europe. But in countries with low middle income countries in Asia, this is a very important pathogen in our NICUs, where a lot of outbreaks have been reported due to Pichia, and this is intrinsically resistant to fluconazole. But more concerning is that now multi-azole and multi-drug resistance, echinocandins, zoles, and amphotericin B has been reported from several countries, from India, from Japan, and all these places. Then the very limited drug of choice is left for treating these kind of yeasts. And earlier, Candida krusei and Pichia kudrezii was given different names, but it's one species. For 4 names have been given earlier, but now the genomic structure have shown that it's one species. And just to give you an overview, that this is a study from Africa, South Africa, where several outbreaks due to rare yeast has been shown. And if you look at the outbreak due to Candida krusei, several times these outbreaks have occurred in the ICUs, then replaced by another rare outbreak. But then again, Pichia kudrezii outbreaks have emerged. And to understand this genomic epidemiology of Candida krusei in ICUs, we did a genomic sequencing study for containers for 5 years in a single ICU, and we found that multiple outbreaks with less SNPs, with the isolates which are highly identical, were identified and picked up. So if we see in the year 2015, we had 3 cases which were highly related. And then in the year 2018 to 20, we had 2 small clusters, one with the isolates in 2018 and 19, and then again in 10 isolates in 2020. So understanding the genomic epidemiology of these outbreaks is also important. And this pathogen is intrinsically resistant to fluconazole. And now coming to these rare species, although not listed, but the local epidemiology is very important. Just to give you an overview of this study, that in neonates, these because of the immunocompromised status, several species will be emerging, have emerged, and this is one of the list of all those rare species which are found from in the neonates, in Greek neonate and children in ICUs are 12-year studies. So we have to be prepared because these yeasts are generally found to be resistant. So if I look at Blanqui outbreak, which we reported, it was resistant to echinocandins. Then Dirychmia churashmensis, it was also intrinsically resistant to echinocandins. We keep on seeing those outbreaks. Lordosmoisis elangisporus had IMICs to fluconazole. So with this, I would like to say that we have to be prepared for several species of rare yeast, which you'll be observing. And I will refer to all of you to this global guideline document. And this global guideline document very clearly gives clear indications how to go with the identification, what samples, the blood culture, what is the guideline. And it also tells you that the identification of these strains it gives you strong recommendations of using Malditoff, chromogenic media, gives you moderate recommendations of using PCR in fish. So this is a good reference for all of you who are attending this webinar. And if you want to see the line of treatment, this is a great document which says the first line of treatment strongly recommended are echinocandins and moderately recommended, and then when to change the CBC and how to follow up, and also the salvage treatment. This I would like to say, I hope I haven't taken enough time. I'd like to say thank you very much and giving me this opportunity to talk here.
Well, thank you, Anuradha. No, you have done well. We are only marginally over
Sorry.
time limit, but that's okay.
How do I remove this? Okay. Stop sharing.
Yeah. So I have a few questions before I sum up everything. And one of the questions for you, Anuradha, is what our colleague Alex is asking, which is chlorhexidine bathing. Would you recommend chlorhexidine bathing to prevent Candida auris transmission out with an outbreak setting?
Yeah. So this is one of the studies from NIH which shows about chlorhexidine bathing, and it shows but higher percentage of chlorhexidine is required to achieve. And also this is a study where I'm quoting that Candida auris still reside in deep follicles, and even after that, although the chlorhexidine bathing showed that it has removed from the surface, but still the Candida auris was inside. So I will say that, yes, that's the basically available, but still repeated testing has to be done again for detection of colonizers.
Okay, thanks, Anuradha. I've got a few more questions, but they're not in particular to one or the other speaker. So, amongst the three of you, whosoever wants to take it up. And one of the things that people have raised, and this was a question that I was going to ask as well, which is about turnaround time. Now, particularly Sevtap, when you were talking about Aspergillus, azole-resistant Aspergillus, then by the time we actually come to know that an Aspergillus in the lab is azole-resistant, there's treatment down the line is a week or 10 days already, and the mortality is high. So what is the solution for this issue? I mean, do you think that we need a baseline data set?
You mean for prevention of the increase in azole resistance in Aspergillus fumigatus?
No. For treatment purposes. Now, by the time we come to know that an Aspergillus fumigatus or whatever is resistant to voriconazole or amphotericin or whichever drug, a lot of time has already passed because A, the diagnosis is difficult, B, the sampling is not always easy, and then if you grow an Aspergillus to actually test for sensitivity, so the turnaround time is not like bacteria. So while the mortality is very high. So how do we reconcile all these in a medical management of patients?
Yeah, to start with, host is a vulnerable host, actually. If a host has developed invasive aspergillosis, so even if with the most efficient antifungal drug, it's really difficult to have a higher efficiency rates in terms of the treatment. I think, again, I will emphasize the need for the surveillance data, because if the rates of resistance is high in your environment, then to start with from the beginning, if it is 10% or more, using a drug which is potentially effective against that azole resistant strain might avoid losing more time. So it might have less effect in terms of the mortality. So surveillance studies are important. Each center and each geographic area should be doing that, since it varies from 0% to very high rates. And if really you have a 10% or more, then your primary therapeutic choice will actually not be voriconazole. After you have the susceptibility data for that specific strain, you can again change your treatment. But to start with, and not to lose time, I think it will be very useful to have your local data.
Yeah, thank you, Sevtap. I agree with what you've said. The many participants have raised this question as well, which is about low and middle income countries, that a lot of data or a lot of suggestions that we make are not necessarily applicable to low and middle income countries, or the guidelines don't exist, or they don't have the facilities for, say, Molotov and also white tech. So what is it that we can do about that?
Yeah, it's really difficult, especially for the identification. With Molotov, we're doing a lot better, of course, in terms of the identification at species level. But for Aspergillus fumigatus that I have focused on, I should say a good basic mycology knowledge, and using the thermotolerance tests, using the microscopic morphology, you can still end up with a good diagnosis in terms of the species to start with. Then on, I think what's more difficult is the susceptibility testing, as I also focused on during my presentation. And especially for that, I believe the screening method that was standardized by UCAST is very helpful, because it's easy, it's less labor intensive, it doesn't need much expertise, and also in terms of the costs, it's more beneficial. So at least as an initial test that might be applied, I suppose, in many of the centers. And then if you end up with a potentially resistant strain at the end of the screening, that might be sent to a reference lab for confirmation. So screening methods are always helpful in that respect, because the microdilution methodologies cannot be implemented in every center.
Thanks, Sevtap. And one final question I had was, somebody raised the issue of salt tolerance test for Candida auris, and I don't know whether Cornelia, you, or whether Anuradha, you want to take it up.
Yeah. So the salt tolerance test can be used for Candida auris, but Candida guillermindi, I mean, we do in our lab. Candida guillermindi also grows high in this, and difficult to differentiate. So it can't be only test for identification, but when you have an isolate which grows at more than 37, 40, 42, and then a salt tolerance can still be used for presumptive identification.
Okay, thank you.
Also Candida tropicalis, Anuradha, probably. Because tropicalis is very virulent and is becoming very resistant. So it's another worried problem.
Yeah. And migration, transfer from one country to another, it never stays there. So the strain which was in China is also now in Indian subcontinent. So yeah, we have to be all the time on our guard and update on the list.
So a very final, Abs, a very final question. I think for all the speakers, probably more Cornelia and Anuradha, some comments on antifungal susceptibility for rare yeasts. So all the yeasts without breakpoints, without anything. So how we have to manage this infection?
Anuradha, start.
So, difficult question, I know.
I know.
But in UCAST, for UCAST, there is an extra document
Exactly,
giving a guidance how to use clinical breakpoints with non-species related breakpoints. Sorry. So you can go to the UCAST homepage. You can download this document and have a closer look.
Yeah, can I also comment on that, Maurizio?
Of course.
As Anu and Connie has outlined, there is a document on a pragmatic approach, how to deal with the rare yeast without breakpoints. And that is based on the idea that these are mostly less virulent than the Candida species because we're having less infections with these. And also based on the similarities in terms of the MIC data that have been obtained so far. So starting with these two ideas, there have been some proposals in terms of how to classify a strain as susceptible or resistant and to start your treatment. So that's why we called it as pragmatic approach, and work together with Professor Michael Arendrup on that. So you can easily have access to that document. This will certainly be useful in terms of the clinical practice when there are no breakpoints.
Thank you, Sevtap. Maurizio, any final comments from you before we wrap up?
No, I think that we try to cover all the spectrum of fungal infections, and also we try to reply to the very, very much questions we received, because I don't know, but I think that we received more than 100 questions probably, because so this of course is fantastic, and now are still connected more than 400 persons. So I think that this my only comment is that this webinar was probably a success, and of course this is because we had fantastic speakers that we stressed very much this important problem. So thank you very much to Sevtap, Cornelia, and Anuradha for this.
Absolutely. Thank you very much, Cornelia, Sevtap, and Anuradha. Cornelia for providing the background, and Sevtap for touching on the Aspergillus resistant isolate, azole resistant Aspergillus, and Anuradha for Candida auris and other Candida, including Candida blancii, which many people wouldn't have known.
Yeah.
Exactly.
Okay.
So the rare yeast will be coming up for sure.
Yes, I mean it's never changed. I mean it's a vast field, isn't it? I was reading the other day about Aspergillus guidelines for museums, because they can damage the artifacts in the museums, and somebody from Denmark, I think, had a guideline on that, or maybe from Netherlands, I don't remember for sure. So it's a very broad field, mycology, and you're all part of it. So thank you very much again to Maurizio, to you,
also from my
and also
side.
to Cornelia, Sevtap, and Anuradha for their excellent presentations. Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Bye.
Thank you for
Bye.
sharing
See you soon.
the session.